There we go. You know, and even as you're all pulling in on this, which I appreciate, I want to kind of note that we probably would never go to like any one thing over any other thing, because I think that's when things get boring, right? So I just kind of want to know, where are we? I'm thinking, you know, did that float pretty well? And then we can continue to mix things up. Even I'm thinking about maybe even mixing things up so crazily when we get to unknown identification that I might just tell you, you don't have to come to lab at any one given time. You could just come anytime during the day and just see how that kind of chaos flies. So we'll see. You know, I just like to mix it up because it's fun for me, it's fun for you, and when we're all having fun, then that's good. So I will bring this back up a little bit later. Thank you for taking time to take this poll, and as I said, more formal feedback will come later. And I'm going to go ahead and jump in with today's announcements. Just a few things. I don't want to, it's Friday, right? I don't want to overwhelm you too much. Yeah, exactly. With anything too logistically difficult. But some of you are still getting that pre-test in. If you want to get those extra points to replace your low homework score, go ahead and work with that. We do have that new homework out there, homework number 2. I already have seen a couple of you with questions on that homework, and I look forward to more questions. I'm super looking forward to the Monday night online office hour because it was so much fun last week. So we'll see you there and hopefully get that homework finished off for Tuesday. Otherwise, we're also looking to firm up the topics. Been loving getting your emails regarding here's what we're thinking about, what do you think about this one versus this one, and sometimes I can give you that scope of having followed the project for longer and knowing which one might give you more information versus which one might give you a more trendy, novel approach. So I'm trying to send feedback on all of those, and I'm loving your emails with regards to that. And in terms of Tuesday's lab, we're just going to do it kind of traditional style on Tuesday, and you'll want to get the pre-lab done for that one. We're going to be doing the epically important gram stain. So this is probably the most foundational lab of the whole semester. Maybe not the most fun one, but probably the most foundational one. And I'd say it probably ranks in the top. You know, well, all of them kind of rank in the top as far as funness goes. So, you know, they're all fun. Let's go ahead and jump in because today we get to cover some really great foundational stuff about the bacterial world and situate the bacterial world within the greater worlds of the 3 domains of life. So by virtue of the 1970s work of a man named Carl Woese, who was a self-proclaimed molecular biologist turned physicist. He did a lot of incredible work on 16S ribosomal RNA sequences and utilizing phylogeny, which is the evolutionary relatedness of organisms, to determine that we could perhaps best divide life taxonomically, which taxonomy is a fancy, sexy word for putting things into boxes. People like to put things into boxes. So we like to say, okay, can we box up life and put it into 3 domains? And so Carl Woese said, yeah, we can do that. We could classify life into 3 domains. And these 3 domains would be bacteria, archaea, and eukarya. Now, of course, we tend to infatuate with the eukaryotic domain because, of course, we belong to that one. And so a bulk of our study tends to be centered in that domain, which is quite ironic, actually, because as it turns out, we are not the dominant life form on Earth, right? It comes as a shock for many people, but in fact, bacteria are the dominant life form on Earth. And I have a beautiful quote, actually, that I want to read to you from Stephen Jay Gould. And he's probably one of my favorite authors. Rachel's nodding. She's read some Stephen Jay Gould. If you haven't, this weekend would be a wonderful time to cuddle up with one of his books. This particular passage comes from a read called Planet of the Bacteria. On any possible, reasonable, or fair criterion, bacteria are and always have been the dominant forms of life on Earth. Our failure failure to grasp this most evident of biological facts arises in part from the blindness of our arrogance, but also in large measure as an effect of scale. We are so accustomed to viewing phenomena of our scale, sizes measured in feet and ages in decades, as typical of nature. Individual bacteria lie beneath our vision and may live no longer than the time I take to eat lunch or my grandfather spent with his evening cigar. But then who knows? To a bacterium, human bodies might appear as widely dispersed, effectively eternal, or at least geological, massive mountains fit for all forms of exploitation and fraught with little danger. Isn't that great? He has a sexy mind. So bacteria by far outnumber all other forms of life. And the interesting thing is that we know very little relatively about them. Less than 1% have been cultured and characterized in the lab. So it is an area of ever-evolving inquiry, and certainly one that we will hope to get you started with by focusing our study on bacteria. Now, does anybody recognize the domain of life called Archaea? Does that one ring a bell? What are they, Christian? Right. We like to perceive them as extremophiles, many of them, though not all of them. But a lot of them do like to live in environments that we consider inhospitable. And we don't think that, say, acid or high temperatures, hydrothermal vents, right, are a particularly great place to live. But Archaea certainly do. They're known as being very deeply branching, which means they're often called the ancient ones. So in that way, they are somewhat unique, though they are a disparate group, and there are members that maybe lie outside of that stereotype. It's interesting because traditionally, what have we called these two domains of life? Collectively, bacteria and Archaea have commonly been called what? Prokaryotes. So most likely, you all at some point learned that life is divided into prokaryotic life and eukaryotic life. Today, I would like to stimulate the question as to whether or not the term prokaryote really has any foundation. And throughout the rest of the semester, I want you guys to ponder whether prokaryote is a term that we should even keep. And today, I'll simply tell you that there are three very distinctive domains of life: bacteria, archaea, and eukarya. Prokaryote is a term that has been used for a long time. We're still going to see it come up in here, but come the end of the semester, I'll probably get you thinking again about whether that question, how we should answer it, right? One quick note, this is totally logistical. The spelling of prokaryote and the spelling of eukaryote can vary. That is, this K can be a C, and that's absolutely accurate as well. And sometimes I use the C. Old habits die hard. So if you see me do that, it's not because I'm stupid, it's because that is also a legitimate spelling, right? I'm not, oh God, some days I'm stupid for sure. But you know, that is a legitimate spelling of prokaryote. So in order to delve into the world of prokaryotes, perhaps, quote unquote, but also really all life forms, we need to learn how to name them. And there's a binomial system of nomenclature. Binomial, of course, bi meaning two, okay? So they have a first name and a last name, more or less. There's a genus name and a species name. The genus name always comes first, and by tradition, that genus name is always capitalized. So the most famous of those is Escherichia. The species name, coli, is always lowercase. And in all cases, we always italicize or underline scientific names. This is something that we will hold you to. You all are going to be doing some scientific writing this semester, and we will hold you to the standards of italicizing or underlining your scientific names. And when you write your genus, we're going to look for that genus name to be capitalized and the species name to be lowercase. It's interesting to think about where these two names derive. And historically, it used to be that people could name bacteria after themselves. And of course, egotistically, they did. And so Theodore Escherich was the discoverer of Escherichia. And how about coli? Anybody see that as a term that looks a little familiar? Colon. It was derived from the colon. I could not for the life of me remember who I was talking to. It was one of the troublemakers of the West Bench the other day about, he was saying, you know, oh, I can't believe I'm touching E. coli. And I was like, well, actually, in your gut right now, you're touching lots of E. coli, right? Because it is a normal flora of our gut. However, there are some strains of it that are pathogenic. So now we come to the third name, right? There's also often a third designation, and that's the strain designation. So often after Escherichia coli, we see something else, like, for example, O157H7. That is a strain you don't want to touch. That one is the very pathogenic diarrheal pathogen that you definitely don't want to contract. It's a very acid-tolerant pathogen. So not one that you want to come into contact to. But most strains are not pathogenic. Okay, so first name, the discoverer often, second name, the location. Now, things have changed a little bit in nomenclature. No longer are people allowed to name bacteria after themselves. So we tend to get some other types of genus names, and that's kind of cool. I'll tell you about a couple here in a minute. So the genus name is commonly abbreviated, and how do you most often hear of Escherichia coli? E. coli, yeah, and it's big capital E period coli. And as I said, we get different strains. Some of them are pathogenic, some of them are not. We work in the lab with non-pathogenic strains of E. coli. Please don't do this, but you could lick a plate of E. coli in the lab, and it would be all right. It would, you know, kind of change up, disrupt your microbiota a little. They'd be like, ooh, this is a new guest, but maybe, you know, a little bit new strain. But it is not pathogenic. We don't work with O157H7. There are some labs on campus that do, and then those are very high biosafety level labs, right? So strain makes a difference. Strain designation makes a difference. Let's now look a little bit about a review of yesterday in lab, and this is going to look super familiar to you guys based on what you saw in your microscopes yesterday. We can have two different basic cell shapes. Who remembers the names of the two shapes, right? We had a circle and we had a rod. What are those called? Cocci, right? So plural, cocci. If you have one of them, it's a coccus. And bacilli, or singular, one bacillus. And we saw both of those yesterday. You guys saw, on your first stain, you saw some bacilli, and the E. coli is a bacillus. And then later, some of you saw unknowns that were cocci. So, for example, I think I was remembering Rachel's, right, had cocci. Yes? Okay. And Olivia, you had, I saw you earlier, you had a bacillus, right? And Marissa, yours was a bacillus, right? So we saw both of these shapes. There are variations, though, within a theme. For example, within the world of cocci, we tend to get different arrangements. So these arrangements, let's write down bacilli. These arrangements are things like diplo, two cells side by side. Who saw some cells on there unknown that were diplo? Two cells side by side. Yeah, that happens. We have some unknowns. For example, unknown number 160 tends to form some diplo. We also see more chains, right? Chains are sometimes referred to as what? Strepto, yeah. And that's because the genus Streptococcus tends to form chains. What isn't true is that all chaining bacteria are in the genus Streptococcus. They're not. For example, Enterococcus forms short chains all the time, but it's not in the genus Streptococcus. However, we might describe it as having the strepto arrangement. So that can be kind of confusing, but that is a thing. So chains. What that means, again, is that the bacterium divides in a single plane rather than in random planes. And those that divide in random planes, for example, those of you who had unknown number 130, you might have seen some staphylo arrangement. That is grape-like clusters. And that is just division in irregular planes. Whereas if you see division in 2 planes, you get a tetrad. Or in 4 planes, you get something that we call Sarcina. So Sarcina is a genus name, but also it is the name of an arrangement. Just in the same way that Staphylococcus is a genus name, and the arrangement Staphylo is an arrangement. But again, Micrococcus, for example, will sometimes form Staphylo arrangement. So don't get confused in thinking that all Staphylo-arranged bacteria are necessarily in the genus Staphylococcus. Am I making sense? Okay, cool. So just so you know, that can be a bit confusing. Bacilli can be different in their shape. There are some that are like rods that are sort of short and fat. Unknown number 140. Some of you saw short, fat rods, and in fact, sometimes your TAs were like, I think that's cocci. And then I would come over and I would say, I think that's a bacillus, a short, fat bacillus. You might even call it a coccobacillus, right? Kind of has that short, fat nature. Somebody asked me about a half-moon shape yesterday. We sometimes call that Vibrio. So a half-moon-shaped bacterium, for example, Vibrio fischeri, one that literally glows in the dark, has a half-moon shape. Spirilla, many aquatic bacteria have this shape. And remember talking about Treponema pallidum, the causative agent of syphilis? It's shaped like a corkscrew. Spirochetes actually move very well through viscous solution, viscous tissue, and they do so the way that your corkscrew moves through a cork, right? They just move just like that. It's pretty cool. So spirochetes, spirilla, vibrios, and coccobacilli are all types of bacilli. But you may not have recognized that, in fact, other shapes can exist. And I was thinking of Courtney this morning, because you're like the literary connoisseur. And I was thinking pleomorphic may be something that has come up in some of your reading. It means shape-shifting. Shape-shifting. So if anybody's like sci-fi, right, these are the shape-shifters, the ones that change their shape. And we'll see later in the semester, actually, we'll work with a bacterium that when it's fresh, it looks like a bacillus, and when it's aged, it looks like a coccus. So it actually morphs over and changes its shape. Some of these guys have very unusual shapes, like, for example, star shapes. We'll see prosthesia, which are like literally projections of the cell that come out and give them a kind of a star shape. So now that we've kind of looked at shapes and arrangements, we can turn our attention and look at the way that bacteria reproduce. Some of you already got a sense for this by looking at your colonies on the plates yesterday. And also look at the size of cells. So recalling your estimation of size on the very first day of lab, who remembers how big, whatever it is that they were looking at, how big was it? You estimated the size on that alga cell or something that you were looking at? Who remembers how big? Anybody? Yeah. Right, 200 micrometers roughly, like maybe took up 1/10 of the field of view on the 100x, something like that. Very large compared to bacteria. And remember how yesterday you had to go all the way to your oil immersion lens? And even so, your unknown bacterium, there were probably 100 or more of them packed across one field of view, right? Making them maybe on the order of 2 micrometers. So we can kind of estimate those sizes in lab, but as a generalization, I can tell you that most bacteria are somewhere between maybe 0.3 and 2 to 5 micrometers in diameter. However, there are some that lie outside of that rule. This is Thiomargarita namibiensis. Want to give a guess where it was isolated? Namibia. It was isolated in 1997 off the coast of Namibia. And what about the genus name? So thinking about the fact that this was really recently, it's no longer the name of the discoverer, but does anybody know what thio refers to? Sulfur. Nice job. So sulfur, this has actually got visible internal sulfur storage granules inside of it. And in fact, this bacterium is so big that you can see it with the naked eye. So obviously, it lies way outside of that generalization. So while we can stereotype bacteria as having roughly this size, there are many things that lie outside of that range. So they're often smaller than eukaryotic cells, but Thiomargarita is certainly an exception to that. We talked yesterday about the fact that bacteria reproduce via binary fission. So that means that they literally elongate and split into 2. They replicate their nucleic acids, put it into 2 separate compartments, and boom, 2 daughter cells. So this idea is termed binary fission, where a single mother cell divides, makes two daughter cells. Those two daughter cells divide, make two more daughter cells. And again, we get that what kind of division? What kind of growth? Yeah, good. I'm hearing some good things. We're maybe getting a peer colony. I heard that because we're coming from single mother cell. The type of math, the mathematical expression for that kind of growth would be exponential growth, right? Because you're going from one cell, that divides into two, to then that each divide into two. So we're getting that exponential growth. I want to watch a quick video. This is on TeacherTube.
Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
And that was what was happening on your plates, right? 5,000 billion bacteria in a single day, in an overnight incubation. So remember how some of you did your on your own and you got way too thick of a smear on the slide? And it was because you're like, man, I got to make sure I get all 5,000 billion bacteria on my loop, right? And in some cases, you did accomplish it, I'm pretty sure, because there were piles, right? Piles upon piles upon piles. So it helps us kind of think about the idea that even if you just barely touch that colony, you're probably getting millions of bacteria, even that you can't see on your loop or your needle. So we're kind of got to get used to the fact that there are so many there, and just seeing them means that we are having quite a lot of them there. So I want to zoom into the bacterial cell, and we're going to spend some time thinking about structure, function of this cell. And so I have a rad model that is pretty simplistic. I think we named it last semester, but I so can't remember the name, so I think you guys should name it this semester. I don't know, Huey or something. We got Humphrey. I like it. Yeah, maybe this is Humphrey. But Humphrey shows us the general structure of a prokaryotic cell in kind of a simplistic way. Some of the things that you notice right off about it. Oh, by the way, Humphrey is what? A coccus or? It's a bacillus. Yeah, it is a bacillus. So Humphrey the bacillus. And we notice that it has inside of it a gel-like mass that is shown in this form as a kind of middle of the cell kind of thing. That's called a nucleoid. That's where all of the nucleic acid of the cell, I shouldn't say all, that's where a majority of the nucleic acid of the cell is stored. We also can kind of see some other things going on in here, little guys. There's going to be some storage granules. We're going to talk about things called inclusions that are holding on to materials that may be used for energy, or they may be used for buoyancy, or just compartmentation in the cell. So we're going to go into that. We also noticed that Humphrey has some like hair-like projections on the surface of the cell. Anybody know what those are called? I know, I get ya. Cilia are only eukaryotic. These are called pili. So they're bacterial pili. And then somebody said flagella. Well, look at that big long black thing there. Humphrey has a polar flagellum. I once had a student dress like that with a polar flagellum strategically placed. Yes, he did get extra credit for Halloween. Okay. So a polar flagellum. So these are some of the structures that we're going to look at in Humphrey. So let's zoom in. And I want to begin with the bacterial cell membrane. So think of us literally taking a zoom-in picture to this structure that's shown in yellow right here, and really looking in depth to that structure. Now, the cellular membrane, as is true in all domains of life, is a selectively permeable barrier. It is distinguished across domains of life by composition of phospholipids and composition of proteins. But what remains the same across all domains is the function and also the dynamics of this structure. That is, it is not a rigid structure. It has quite a bit of flexibility within it. So we'll come back to that model and that notion, but I want to review the labeling. Remember how I told you that always on my pictures, exterior of the cell is up. So our model, exterior here, interior here, and that's always up and down. So it's almost like we're looking at the top membrane on our cell model. Looking at the proteins and the phospholipids that comprise this membrane, we can give some names to them. The ones that span the membrane are called integral membrane proteins. They pass through the whole membrane. And I think we talked a little bit about some of the functions that those might have, and we're going to review that in a moment. Now, we've also got peripheral membrane proteins. These are the proteins that don't span the membrane, but instead they just sit next to it and perhaps are associated with an integral membrane protein or just associated with the polar heads of some of the phospholipids. We also see some other structures in bacterial cells that may not be familiar to you. These are called hopanoids. But perhaps when I tell you that hopanoids modulate the fluidity of a bacterial cell, they keep it from freezing when it gets too cold, they keep it from melting when it gets too hot. What does that start to remind you of? Cholesterol. Yeah. But guess what? Where is the only place that we find cholesterol? Animal cells. Yeah, good. Mammals. Good. And so we don't see cholesterol in bacteria. And so in bacteria, the functional equivalent is the hopanoid. It has that function, but it is not it's not exactly the same as cholesterol. It's interesting to know that hopanoids are thought to contribute quite massively to oil reserves, so fossil fuel reserves, because hopanoids are a very prevalent molecule in the most prevalent form of life, right? So that's an interesting thing to think about, them being a lipid, right? A lipid store in that way, a fuel store. Notice that this particular lipid is attached to what? If we call it a glycolipid, it's attached to carbohydrates, right? So this glycolipid has carb and lipid, two macromolecules. What would we call a protein that had a carb region on it? Glycoprotein. So we do see a lot of glycoproteins as well. In fact, much more in the way of glycoproteins than we used to think there existed in the bacterial world. So let's talk about that model then, by which the membrane is not static and rigid. It is very fluid and dynamic, right? So I had a student some time back that was awesome. She remembered the fluid Mosaic model by thinking about a membrane as country line dancing, because you're allowed to move laterally and rotate, but you don't flip-flop, right? You don't do cartwheels. And so at least I don't think most people do, but maybe you do. But if you do, then go with it, rock that. But our membrane proteins don't, right? They don't flip-flop unless they have some sort of help. So those are the motions that are allowed. The lateral diffusion and rotation, but no flip-flopping, okay? And that is the fluid Mosaic model. It's interesting to note that the root of that term, right, fluid, is pretty obvious, right? Meaning that things are, phospholipids and proteins are rotating and diffusing laterally, not flip-flopping. But the Mosaic part comes from the fact that you've got this semi-permeable phospholipid bilayer with interspersed proteins in it. And those of you who are art buffs will think, it's like a Greek mosaic or a Roman mosaic. And that's the pattern. The proteins are interspersed like a mosaic throughout the lipid bilayer. Excellent. Yes, please. Please.
The phospholipids, they can go in any direction.
Only lateral and rotation. What is required? Let's make this a little deeper and sexier. What is required to enable them to literally pull? Like, think about this, right? Pull their polar head group through the hydrophobic tails and over to the other side. You think that's going to be very energetically or thermodynamically or anything favorable? No. So what's going to be required? What kind of protein might catalyze that? Enzymes, right. Enzymes are needed, and get this, the name of them is flipases. They are called the flipases, yeah. So it requires an enzyme in order for that kind of thing to happen, Brent, yeah. Great question. Hey, so I made a lame animation, but it kind of gives you the idea that, oh no, it's not playing. I'll find a way to do it. It gives you the idea, I know that's how lame it is, right? That they are not rigid, right? That there is a fluidity to them. So here we go. Toad's going to get this. Now, what my animation doesn't show is it's hard to tell the rotation is happening, right? You can't really see that rotation. But I hope that it gives you an idea that a membrane is not rigid and inflexible, that it is very dynamic. So then the questions that I want to ask you guys to think about that relates some of our earlier coverage is, would the amino acids found in the membrane-spanning region of an integral membrane protein love or hate water? So let's think about what type of amino acids you might see in this pink integral membrane protein spanning this region, maybe sticking their R groups out right here. What kind would those be? Hydrophobic. So give me an example. What might be an amino acid that would stick its R group out right there? Luke's got his table. Go. Okay, maybe glycine's not super water-loving. It's the little flexy one, right? It likes to fit into small places. But what would be really water-hating, Stan? Thank you. You were totally called out just then. Yeah, so Stan says phenylalanine. What's another one? Phenylalanine's very water-hating. Leucine. Isoleucine, right? All of those big-time hydrocarbon R groups might be found there sticking their heads into the nonpolar phospholipid bilayer. Good, excellent job. How about here? What about sticking their R groups out here into the aqueous exterior? What might be an example? Yeah, lysine, excellent, good. Or, you know, maybe aspartate or glutamate, right? Some of these big-time water lovers. Excellent, Lauren. How about this? Would it be easier to extract a peripheral membrane protein or an integral membrane protein? So what if we wanted to isolate this pink guy as versus a peripheral membrane protein that might be, where did you go? Oh, okay, problems. Versus a peripheral membrane protein that might just be associated right here on the outer leaflet of that membrane. Which one would be easier to extract? Yes, right, easier to extract. Harder to extract an integral membrane protein, right? Because you would actually have to solubilize the membrane. And that would mean, literally in a pragmatic sense, that you would have to add detergent in to dissolve the membrane, right? Like dissolves like. Anybody know what detergents look like? They're polar and nonpolar, means they're also, what would we call them? Amphipathic, just like the phospholipid bilayer. So they like to dissolve it, and that would actually free the integral membrane protein. So that would be the pragmatic way that you would actually free that integral membrane protein. It's much harder than getting a peripheral membrane protein. Okay. All right, okay, good. So one more question, I guess, or two more? No, one more. What are some functions of an integral membrane protein? We reviewed some of them. Transport proteins, right? So maybe letting things into the cell or out, and sometimes they're active transporters, sometimes they're passive transporters. We also talked about them being receptors, good, so for signaling purposes. And then lastly, what is one other function that we sort of ended off with last time? This idea that some of these move electrons, and they are part of the electron transport chain. Excellent. So that's actually where I want to take our coverage next, but not before we do a little bit of definition regarding, oh good, it's playing again, regarding the process of simple diffusion versus transport. So I want to just make sure that everybody understands what I mean when I say selectively permeable, because I was throwing that term out with wild abandon. And what that means is that essentially the membrane of a cell is kind of like a bouncer at a Soup's Popular Dance Club. Because remember, bouncers at super popular dance clubs are scanning the crowd, they're looking around, and they're like, okay, ladies with low molecular weight, you're in, you're in, you're in. And ladies that don't want to drink water, hell no H2O, right? You're in. Okay, this is exactly what a membrane does, right? What kind of molecules can get through without any kind of charge? I mean that like in parentheses. Like what kind of molecules pass freely across a membrane? Small, low molecular weight, and hydrophobic. Hydrophobic, good. Yeah, so non-charged would be another way to say that, Bridger. So things like oxygen, carbon dioxide, they pass by simple diffusion. Everything else requires a transport protein. If it's charged, it's going to have a transport protein. If it's large, it's going to have a transport protein. But those small hydrophobic molecules, they're going to get right in. So I want to talk a little bit about those particular integral membrane proteins that are involved in the electron transport chain. So prokaryotes that are capable of respiration or photosynthesis, they're going to be equipped with members of the electron transport chain. And remember, as we ended our hour last time, we were talking about the fact that a cell can rip and strip high-energy electrons, take them to the ETC, pass them through the ETC, and they utilize the energy that comes from those high-energy electrons to do what? We finally got there at the end of the hour. Create a proton motive force. Excellent, Christian. So creating a proton motive force, and that proton motive force can be tapped into to do what? Create ATP, right. So this is what the central function of an electron transport chain is to transfer electrons through members, many of which are integral membrane proteins, to eject protons. And you chemical engineers are going to love this. It creates something that is sort of equivalent to an electromotive force, except for that it's a proton motive force. It is a pent-up battery-like power that can be converted from that form of pent-up energy into mechanical energy in a machine called ATP synthase that literally turns and exchanges ADP for ATP and makes ATP. So this is a nice process of the first law of thermodynamics, or energy forms getting converted from one form to another. Now, what kind of respiration is this cartoon bacterium using? That is to say, as these electrons are transferred through the members of the chain, eventually at the very end of the chain, the last member in this electron relay team is oxygen, right? It is the terminal anchoring leg of the electron relay team. It's carrying the baton home. Oxygen is the best acceptor of electrons. It has the highest affinity for electrons. So this particular organism, in fact, this is a picture of E. coli's electron transport chain. E. coli uses what kind of respiration? Aerobic, right? Because it is using oxygen as its terminal electron acceptor. So here we're seeing an aerobically respiring bacterial cell, and across its membrane, so out here, it has formed a proton motive force that it can tap into and use to make ATP. What's really interesting to note is that that pent-up proton motive force is not only used to make ATP, but it can be used for other things as well. So for example, with Humphrey, flagellar rotation, right? So when PMF rushes in, one of the things that it can power is flagellar rotation. It can also power transport. Some molecules follow the PMF in. So hopefully everybody's seen Finding Nemo. You remember that scene where the turtles are riding the EAC, right? Okay, well this is very much what many molecules do across the membrane. They ride the PMF. So the PMF serves as a sort of downstream river-like gradient that they can ride into the cell. So the PMF is not only used to make ATP, it is also commonly used to propel flagella, to get flagella rotating, and it's also commonly used to transport other molecules in on the backs of that PMF pent-up battery force. Now, I promise you that an underlying theme in all of our classes from here on out will be the electron transport chain. But does anybody have kind of basic, wow, Rachel, you lost me, like 10 minutes ago questions, because I know it's Friday. Okay, pretty cool, Brent?
Kind of a comparison question.
Yeah.
So in eukaryotes, all of the interaction of energies done in the mitochondria, is this bacteria or other
Mm-hmm.
well, bacteria?
Yeah.
They don't have that. Do they just inject protons
Yeah.
in and outside of their cells?
Yeah. So this is actually a phenomenally awesome question to segue into evolutionary theory, and hopefully hit on something that all of you guys understand. That is, this bacterial cell does not have mitochondria. Okay, so Humphrey doesn't have any classical internal true membrane-bound organelles. We'll talk about some of the debate there, but it doesn't have mitochondria. And we know in us, right, mitochondria are the powerhouses of our cell. They are the organelle in which a proton motive force is formed, in which ATP is made from that proton motive force. So bacteria lack those. Because of that, bacteria, well, and this is kind of like a because of, that's probably another chicken and egg argument. Bacteria do not have mitochondria, so their gradient, their proton motive force, is across their cellular membrane. So the PMF is literally established outside of the cell. So the proton gradient is outside here. So help in relating that to eukaryotic cells. If we say that a mitochondrion is roughly 0.2 to 5 micrometers, a mitochondrion is often a bean-shaped organelle. A mitochondrion has an inner and an outer membrane. We're going to talk about that later. What do you think? Mitochondria came from bacteria, right? This is the endosymbiont theory. So in fact, bacteria, right, are sort of the ones with the novel idea, and they are generating that PMF across their cellular membrane much in the same way that the organelle mitochondrion does, right? Most likely, right? Many evidences to show that that derived from a bacterial cell. Does that make sense? Yeah, it's a great question, Brett. Okay, cool. So that being said, I saw a lot of you guys photographing the screen, which means this picture must not have printed, so photograph the screen real quick. I have a, you want to do it? Okay, I'll go back. Fail. Take a picture. Quick. What did we do before smartphones, right? Oh, did you get, I mean, my 3 to 5 section last night realized that if you took the, taking the picture through the microscope lens, if you took it with Snapchat, it came out better, it was easier, right? At least for some people, yeah? Then you figured out how to save them, right? Yeah, okay, cool. Any other really awesome, sexy-minded questions like Brent's about the proton motive force and ETC and making ATP from it? William?
Is that why cells have like a negative net charge?
Yeah, okay, great. So William's question is another fantastic one, and that is to say that overall, a cell has what we term a delta psi and a delta pH. Overall, giving it that proton motive force. The delta psi is an actual electrical difference in charge. So we could say that overall, with respect to charge, cells, well, bacterial cells, will have a positive charge outside, a negative charge inside, and it does actually create that delta psi. So yes. Now, there are other reasons for charge gradients in human systems that I'd love to chat with you about sometime. But this is absolutely the case. And also delta pH, right, a difference in pH, it is more what? Like the pH is lower or higher outside of the cell? Lower, right, a lower pH outside of that cell because the protons are getting pumped out there. Well, that's one of the ways that was used to actually to support the hypothesis that a proton motive force could be used to make ATP. That wasn't done until the 1960s. It took a long time for a man named Peter Mitchell was able to show that that was the way that this PMF was being used to make ATP. Very good question. So let's see, I want to ask you one now. Which statement is false about the following prokaryotic cell plasma membrane? A, the protein labeled B is a peripheral membrane protein. B, according to the fluid mosaic model, the protein labeled A is free to diffuse laterally and rotate. According to the fluid mosaic model, this membrane is rigid and flexible, and it has a lot of sterols as a primary component. D, the protein labeled D is a glycoprotein. And then finally E, although not pictured here, many bacterial membranes contain hopanoids, which play a role in modulating membrane fluidity. It's a pretty easy question. What's our answer? Yeah, right? Definitely not rigid and inflexible, but very dynamic, and a lot of line dancing going on here. Okay, all right, cool. So looking into the cell itself, we're going to talk next about the inclusions, which are these sort of sometimes storage, sometimes buoyancy molecules. They range in their overall purpose, but inclusions include structures that are granules, crystals, and globules. Those are terms given to various inclusions. So these are often used to store particular molecules. And if we think about situating that within our knowledge of macromolecules, we could think about the kinds of macromolecules that might be stored in these inclusions. So two different kinds, they're either unbound, where it's just a discrete kind of succinct region of storage, or in some cases, they're bound by a single monolayer. That is, if we thought about that, rather than a true phospholipid bilayer, some of these inclusions actually do have around them a monolayer of phospholipids. So that would be like having just one leaflet, right? One leaflet only, and not a true bilayer. So what are some of the macromolecules that a bacterial cell might want to store in these inclusions? What might behoove it or give it a good boost? Carbohydrates, right, good. So carbohydrates might be stored in there. For example, there are glycogen granules. So remember, that is the polysaccharide of glucose with branches on it. Lots of glucoses, all bound together by what kind of linkage? Glycosidic, right? Lots of glycosidic bonds holding together those glucose molecules. The other thing that you might see are polyhydroxy butyrate. Now, some of you might recognize this is another carbohydrate. It is another means of energy storage. It is another polysaccharide. So the poly kind of gives that away, different than glycogen. And we also may see polyphosphate. Now, that being said, can anybody think about why it might be beneficial to a cell to have literally lots of phosphates all bound to one another? So for you chemistry folks, that's a bunch of PO4 3 minuses, although they'd be 2 minuses, bound to one another, right? Or for you more bio folks, that's like inorganic phosphates like PI, 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 all bound to one another. What might that be used for in a cell? Energy, because in and of themselves, those are high energy bonds. Because how does one negatively charged phosphate feel about a neighboring negatively charged phosphate? Bad, right? They repel one another, meaning that makes the bonds holding them very high in energy. They're very high energy bonds. So in and of itself, polyphosphate is very high in energy. But also, what could it be used to make? Sure, good, yeah. So it could be nucleic acids or also ATP. Right? Energy currency. So we can see those being tapped into for a lot of reasons. These are commonly given the term metachromatic or volutin granules. We also see cyanophycin granules. These store aspartate and arginine. And a quick look at particularly arginine might give you the feel that these are important nitrogen storage granules. And of course, fixed nitrogen, a source of nitrogen to build macromolecules from, is very important in any cell. So this storage of that nitrogen-rich amino acid, particularly arginine, is good for storing that which can be used to help build macromolecules. We also can see microcompartments, and this is where I mentioned that the argument about bacterial cells not having true membrane-bound organelles gets a little nebulous, because although they don't have, these are not contained by a phospholipid bilayer, they are actually contained by a protein capsid. In many cases, it is a polyhedron type protein capsid. But what's really crazy about microcompartments is their functionality. For example, one called carboxysomes actually literally fix carbon. So that is a sexy way to say what process is happening in there. What is the fixation of carbon? Fixing carbon. Yeah, good. So, Christian, right? Christian's right on. Photosynthesis, what does that seem functionally equivalent to in a eukaryotic system? Not us, but yeah, in plants. Chloroplasts, excellent, yeah. So this is where things get a little nebulous because these were relatively recently discovered, and carboxysomes seem to form a functional equivalent to chloroplasts and their ability to actually be locations of carbon fixation. Does anybody else want to throw out a sexy-minded something that's going through your mind? Andre's back there. I know he's pondering something. We've got these outer capsids that are icosahedral. They're like a protein coat. What does that remind you of? Yeah, I'm planting the seed. What about viruses, right? Viruses are basically nucleic acids with a protein coat around them. And it's really weird to think about that. I just will plant the seed. There's some interesting literature, and it's contentious, but I'm just going to let you think. I'm going to let you think, that's all. Yeah. So other inclusions that I'll leave you with as you leave today, these are magnetosomes. Magnetosomes are literally packed with iron sulfide and iron oxides, like magnetite and fool's gold. Iron sulfide is fool's gold, right? Rad stuff, iron pyrite, right? Yeah, so guess what? These magnetosomes literally allow certain bacteria to orient with respect to the Earth's magnetic field. So they actually move around based on magnetism. All right, have a fantastic weekend, you guys.